Hi there. Welcome to the first of 2 lectures on the gastrointestinal tract. Let's get started with a brief overview. The GI tract extends from the oropharynx to the anus. Think of it as a long tube whose primary function is digestion and absorption, as well as an immunologic protection. The different organs of the alimentary system serve different purposes and as such have different functional components. However, the general structure of the walls of each organ can be broken down into 4 layers: the mucosa, the submucosa, the muscularis externa, and the adventitia or serosa. The mucosa is a mucous membrane that can be further subdivided into 3 layers: a lining epithelium, the type of which will depend on which organ we are in, the lamina propria, which is a layer of loose connective tissue, and the muscularis mucosa, which is a layer of smooth muscle. Deep to the mucosa is the submucosa. The submucosa is a layer of dense, irregular connective tissue that contains blood vessels, lymphatics, and nerves. This layer may also contain glands depending on which organ we are in. The next layer we see is the muscularis externa, which consists of 2 layers of muscle, or 3 layers in the stomach. The orientation of these 2 layers of muscle is crucial. They are arranged perpendicularly as an inner circular layer and an outer longitudinal layer. This organization is crucial for the peristaltic action of the GI tract. Note that in the majority of the GI tract, we see autonomically innervated smooth muscle. The exception to this is the upper part of the esophagus, where there is skeletal muscle, but we'll get to that in a few minutes. The organs of the GI tract are covered by either a serosa or adventitia. Adventitia is a connective tissue layer that covers organs that are attached to the body wall. Serosa covers the rest of the organs and is comprised of a simple squamous epithelium, the mesothelium, and connective tissue. This image is a cut image from the esophagus. We'll come back to it in a few minutes, but let's use it for a second to look at those layers that we just discussed. Here we can see the mucosa, made of the epithelium, the lamina propria, and the muscularis mucosa. Next we see the submucosa, along with an associated gland. We can also see the muscularis externa here. Now that we have a general overview of the alimentary system, let's dive a little deeper, starting in the oral cavity. The salivary glands open into the oropharynx and are comprised of secretory acini and ducts. The acinar cells are surrounded by myoepithelial cells and can be further divided into mucus and serous cells. The aptly named mucus cells secrete a thin, watery mucin, and the serous cells secrete a mixture of water, electrolytes, and alpha amylase to begin the process of digestion. We can distinguish between the different types of salivary glands based on the proportion of mucus to serous cells. The parotid gland is primarily comprised of serous cells. The submandibular gland is comprised of both serous and mucus acini, and the sublingual gland is comprised primarily of mucus acini. The ductal system is responsible for modifying the composition of saliva, which is initially similar in composition to extracellular fluid. The saliva is modified primarily by the intercalated and striated ducts. The intercalated ducts secrete bicarbonate and absorb chloride, while the striated ducts secrete bicarbonate and potassium and absorb sodium. Let's move into the esophagus, whose primary function is to move food from the mouth to the lower esophageal sphincter via peristalsis. The esophagus consists of the same 4 layers we discussed previously, but let's talk about what's specific to the esophagus. In the esophagus, we have a simple squamous non-keratinizing epithelium and submucosal glands. Let's talk specifically about each of those 4 layers. So as I mentioned, we have our simple squamous epithelium here. From about here to here, we have our mucosa, and from about here to here, we see our submucosa. Next, we have our muscularis externa. Recall that we have 2 layers of muscle here. We have an inner circular layer and an outer longitudinal layer. These 2 layers will appear different based on what kind of section we're looking at. And lastly, we have our adventitia and serosa. The esophagus has a unique muscularis externa, as it is the only place in the GI tract where we will see skeletal muscle in this layer. The length of the esophagus can be broken into thirds. The upper 1/3 consists of skeletal muscle. The middle 1/3 is comprised of a mix of skeletal and smooth muscle, and the lower third is comprised of smooth muscle only. We'll now move into the stomach, which is made up of 3 parts: the cardia, the fundus or body, and the pylorus. As with the esophagus, we have the same general 4 layers previously discussed. At the gastroesophageal junction, we see a stark switch from the stratified squamous epithelium of the esophagus to the simple columnar epithelium of the stomach. This junction may also be referred to as the squamo-columnar junction. The mucosal epithelial lining of the stomach is comprised of surface mucus cells. Think about what the purpose of these cells might be. Here, we also see longitudinal folds of the mucosa and submucosa called rugae. These rugae allow the stomach to expand when filled. The mucosa of the stomach is unique in that we see invaginations called gastric pits. These pits are lined by specialized cells and also provide openings for several gastric glands present in the lamina propria. Let's focus on 4 of these specialized cells: the parietal cell, chief cell, mucous neck cell, and enteroendocrine cell. The parietal cells are these large cells that can be seen here, and at higher magnification can be seen here. These cells are responsible for secreting hydrochloric acid and intrinsic factor. Note the spherical nuclei and eosinophilic staining. The cytoplasm of these cells stains with eosin due to the large number of mitochondria. These cells also contain extensive intracellular canaliculi, which communicate with the lumen of gastric glands. Mucous neck cells are similar to the surface mucus cells lining the mucosa in that they secrete mucus. However, they are different in that they are smaller cells located in the neck of the glands, and they secrete a thinner, less viscous, soluble mucus under vagal stimulation. This is in contrast to the thicker, bicarbonate-rich, insoluble mucus that is constitutively secreted by the surface mucus cells. Chief cells can be seen here, deeper in the gastric glands than parietal cells, and here at higher magnification. These cells secrete pepsinogen. Note the basophilic staining of the cytoplasm with the numerous rough endoplasmic reticulum on the basal aspect of the cell and the zymogen-filled secretory granules in the apical cytoplasm. The last type of cell we will discuss is the enteroendocrine cell. These cells require special staining and are responsible for hormone secretion. More specifically, they secrete gastrin, ghrelin, somatostatin, and histamine. As a side note, you may see mitotic stem cells present in the crypts of these gastric pits. These are responsible for the frequent regeneration of the epithelial lining of the stomach. The submucosa of the stomach is comprised primarily of dense irregular connective tissue. At the border of the mucosa and submucosa, we see GALT, or gut-associated lymphatic tissue. Deeper into the submucosa, we will see blood vessels, the autonomic nerves, and the ganglia of the submucosal plexus. As I alluded to earlier, the muscularis externa of the stomach is made up of 3 layers of muscle as opposed to 2. As with the esophagus, we have an outer longitudinal layer and inner circular layer, but here we have an added middle oblique layer. And finally, the stomach is an intraperitoneal organ, which means it is entirely surrounded by the peritoneum, or the serosa. Now that we've gone through the 4 layers of the stomach, let's move into the small intestine. We'll talk about the small intestine in 3 parts: the duodenum, jejunum, and ileum. The duodenum receives chyme from the stomach as well as pancreatic secretions. The combination of these 2 allows for the breakdown and absorption of nutrients. Here we see evaginations. These finger-like projections are called villi, and each villus is lined by microvilli. Think about the significance of these microvilli and what they might mean for the absorption of nutrients. Each villus contains a core of lamina propria with blood vessels, a central lacteal, smooth muscle cells, and immune cells. We also see invaginations of the mucosa into the lamina propria as we did in the stomach. These short tubular glands are called crypts of Lieberkühn. The enterocytes that line the villi are absorptive, while those that line the crypts are secretory. Stem cells are located at the base of the crypts, which replenish sloughed off enterocytes. In the duodenum, you will also start to see mucus-secreting goblet cells. If we zoom in, you can see the pale staining cytoplasm of these unicellular glands. These cells will increase in number as we move from the duodenum distally. The submucosa of the duodenum notably contains Brunner's glands, which can be seen here as well as here. Note that this is the only place in the intestines where we will see submucosal glands. Other intestinal glands will be located in the mucosa. The secretion from these glands is high in bicarbonate to neutralize the acidic chyme entering from the stomach. Enterocyte function is regulated by the enteric branch of the autonomic nervous system. More specifically, it is innervated by the submucosal plexus, or Meissner's plexus, which can be seen right over here. The muscularis externa is comprised of the same two perpendicular layers of smooth muscle we have previously discussed, an inner circular layer and an outer longitudinal layer. The peristaltic action of these muscles is controlled by the enteric division of the autonomic nervous system as well. Right here, we can see the myenteric plexus, or Auerbach's plexus. This plexus is responsibility for gut motility. And finally, because the duodenum is mostly retroperitoneal, it is covered by adventitia as opposed to serosa. The jejunum has the same general four-layer structure as the duodenum, with some notable differences. Both the wall and lumen of the jejunum are thinner than those of the duodenum. We also begin to see folds of the submucosa called plica circularis. These are responsible for increasing the surface area of the overlying mucosa. Unlike the duodenum, the jejunum is intraperitoneal and is therefore covered by a serosa instead of adventitia. The ileum is even thinner than the jejunum, but again has the same four-layer structure and plica circularis that we just saw. Note these distinct round entities in the ileum. These lymphatic nodules are called Peyer's patches. The epithelial cells covering these Peyer patches contain specialized enterocytes called microfold cells, or M cells. These are antigen-presenting cells that transport antigens from the lumen of the intestine to antigen-presenting cells, which can then initiate an immune response. In the ileum, we also see enteroendocrine cells and Paneth cells. Paneth cells secrete alpha-defensin and lysozyme and are easily identified by their highly eosinophilic staining granules. These antibacterial compounds help maintain the gut flora. Like the jejunum, the ileum is intraperitoneal and is covered by a serosa. We are nearing the end of our journey as we enter the colon and rectum. You've heard me say it before, but I will say it one more time. The colon and rectum follow the same general four-layer blueprint that we've been using this whole time. Like the small intestine, the colonic mucosa has many tubular glands, or crypts, that extend into the lamina propria. Unlike the small intestine, however, there are no villi present. Most nutrients have been absorbed by this point, and the primary absorptive function of the colon is reabsorption of water and electrolytes. Here the mucosa is lined by enterocytes and an even larger number of goblet cells than we saw in the ileum. Recall that these can be identified by their pale staining cytoplasm. Note how many we see here in the colon. In the lamina propria, we again see immune cells and GALT. The submucosa of the colon contains dense irregular connective tissue and has the same plica circularis we saw in the small intestine. The muscularis externa of the colon is unique. While it is still comprised of two layers of smooth muscle, the outer longitudinal layer is thin, with thickened bands running the length of the colon called tinea coli. The myenteric plexus can also be seen between these two layers of smooth muscle. The rectum is similar in structure to the colon, the notable difference being that the outer longitudinal layer of smooth muscle in the muscularis externa is relatively thick. Alright, my friends, that is the end of our journey through the GI tract. In the next module, we'll be discussing the hepatobiliary system, but for now, as a study tip, I highly recommend making yourselves a table like this and filling it in with the unique features of each part of the GI tract. Try and connect these features to the functional physiology of each organ as you go.